and cell biology. His research interests include biochemical and molecular aspects of B12 and folate absorption, transportation, and metabolism, genetic abnormalities in these pathways, and the cellular and metabolic consequences of B12 and folate deficiencies. He's looked at B12 and folate and homocysteine metabolites in elderly population with cardiovascular disease and cognitive disorders, including Alzheimer's, and looked at the neuropathology of these disorders and its effect on the brain. Active areas of research in his lab include, and are most important to autism, the folate receptor autoimmunity, how it relates to both neurotube defects and cerebral folate deficiency and autism. His research focuses on fetal and neonatal brain developments and the role of folate and B12 in this process. And he has really been the one that found the folate receptor alpha autoantibody, which I think is really an extremely important finding that will help us therapeutically in helping to improve the lives of children, both by prenatal treatments, improving neurotube defects, the development of, or preventing the development of neurodevelopmental disorders, and autism. He holds a bachelor's in chemistry from the University of Hunan, master's in applied biology from the University of Bombay, and a PhD in biochemistry from the University of London. So with that, I will leave it to Ed to give you a fantastic talk.
Thank you, Richard, for that great introduction. It's a pleasure to participate in this conference. Let me share my screen with you. Should I hit the full screen? Okay, good.
Yeah.
Okay. Right. I'm trying to see if I can. I'm seeing you, Richard. Can I see me?
I can see your slides.
No, but can I? Where am I? I don't see myself.
Oh, I don't know.
Okay.
I can't see you.
Okay,
But
all
I
right.
can see your slides.
I can see many people here. Okay. All right. So today I'm going to talk about folates, folate receptor autoantibodies, and the connection to autism spectrum disorders. In terms of disclosure, I would like to mention that the patent for the measurement of folate receptor autoantibodies is owned by the university, the research foundation, and I'm going to talk a little bit about the off-label use of folate or folinic acid in this case. So what is autism? We know autism as a behavioral disorder, and it is pretty evident that it is as a result of disordered brain development during fetal life and early childhood. The causes of autism remain unresolved. Consensus dictates that it is a multifactorial disease. That means it is not a single gene disorder. It does not follow Mendelian inheritance, and it doesn't follow typical Mendelian genetics. It is multifactorial, and those factors remain unresolved. Now, what is folate, and how does it connect to brain development and function? This is what I'm going to discuss today, and ask a rather reactionary question: like could a single nutrient contribute to the clinical phenotype of autism? Okay, now just to introduce folate to the audience here, folate is a generic term used for vitamin B9. Folic acid, as we know, seen in preparations of multivitamins and other B complex vitamins, is a pharmaceutical preparation that does not exist in the body, but can be readily converted to metabolically active form. It's used in pharmaceutical preparations because it's a very stable form and it can be readily synthesized. Now, normally during absorption, folic acid is rapidly converted to methylfolate due to the high dihydrofolate reductase and methylene tetrahydrofolate reductase activity in the gut. So at physiologic concentrations, when folate is introduced in the diet or orally, most of it will be converted to methylfolate before it reaches to circulation. Therefore, methylfolate is the major circulating form of folate and is taken into cells by the folate receptor alpha or the reduced folate carrier. Under physiologic conditions, it is primarily the reduced folate, the folate receptor alpha that functions, which has a very high affinity both for oxidized form as folic acid or as a reduced form of methylfolate. The reduced folate carrier, on the other hand, does not transport folic acid, but it can transport methylfolate and other reduced forms of folate. However, it needs a high local concentration for it to be activated and transported. All of the metabolically active forms are generated from methylfolate via the enzyme methionine synthase, which is a B12-dependent enzyme, and the dihydrofolate reductase enzyme. Okay, so before you get overwhelmed by this, let me simplify this diagram for you. And what it shows is basically all of the single carbon exchange reactions and metabolic pathways involving folate. So folic acid is converted by dihydrofolate reductase to tetrahydrofolate, and that tetrahydrofolate pool then forms all of the reduced forms of folate that are actively involved in transporting single carbon units or carbon skeletons for the synthesis of various compounds. Among them, most important is the synthesis of purines and pyrimidines, which contribute to DNA and RNA synthesis and ultimately to cell replication. It is also involved in production of methionine via the homocysteine methionine conversion, and this requires vitamin B12. And the methionine formed is an important precursor for maintaining intracellular SAM, or S-adenosylmethionine, which is considered a universal methyl donor for methylation of DNA, RNA, and proteins. Okay. The homocysteine there is also a contributor to cystathionine production, which is very high in the brain, by the way. And cysteine produced from there is a source of cysteine for glutathione production, which is important for maintaining the so-called cellular redox state and mitochondrial function. On the other hand, if you look on the left bottom side here, one of the purines produces GTP. GTP is hydrolyzed by the enzyme GTP cyclohydrolase to produce tetrahydrobiopterin, which is the important cofactor for the hydroxylation of aromatic amino acids to produce neurotransmitters. So as you can see from this diagram, that there are so many pathways and so many reactions that can basically control cell replication and metabolic reactions. So it could have a profound effect on brain development and function, as you see in fetal as well as neonatal brain development. So to summarize, since folate has a major role in purine synthesis that leads to DNA synthesis, and methylation reactions that leads to regulation of gene expression. This could profoundly affect neural development, and that in turn could affect brain function. And hence the connection between folate and developmental disorders. So looking at folate, there are many ways one could develop folate deficiency, whether it is malabsorption, low dietary folate, or genetic defects of folate pathways. And more recently added to this repertoire is the folate receptor autoantibody that could impede the function of folate transport. Folate deficiency, on the one hand, as seen on the left here, could lead to various pregnancy-related disorders. That includes infertility, miscarriage, preterm birth, neural tube defect pregnancy, and other gestational disorders. Shown on the right-hand side here is what could potentially happen after birth to the developing brain. Here you could have effect on metabolic processes, DNA RNA synthesis, again, the methylation and epigenetic regulation, effect on neurotransmitters, neurogenesis. Among them, what is seen and identified in autism is the cortical layering defects, axonal connections, and dendritic branching and pruning. So one can envision a scenario where basically so many different things are affected that not one single pathway or cause could be attributed to what we see in autism spectrum disorders. So just to introduce folate receptor autoimmune disorder here. Folate receptor autoimmune disorder presents with blocking and binding antibodies in the blood, which can be readily measured by standardized assays. What we know so far is that approximately 70% of the children with autism spectrum disorder are positive for both either blocking, binding, or both of these antibodies. These antibodies are prevalent in families with ASD children and ASD patients. What we know so far is about 38% of the parents are also positive for these antibodies. What is really interesting and fascinating is that these antibodies are also prevalent in unaffected siblings. Now, the main question here is, why are these children not affected? And this is a major topic for discussion, which I would not go into here, but just to indicate that there could be multiple factors that protect one fetus from the effects and another fetus is affected. So this could happen under various circumstances, and we could discuss this at a different conference later. Now, the presence of folate receptor antibodies causes these neurologic deficits associated with autism spectrum disorder. But how severe the neurologic defect is and the onset of the defect is dictated by the appearance and the titer of antibodies. So to show here, if at a very young age the child develops antibodies, this could lead to a very severe form of developmental deficit, such as the CFD syndrome. As the child develops antibodies later on between years 2 and 5, it could lead to spastic ataxic syndrome, intellectual disability, as well as learning deficits. And if the child develops later on between the age of 5 and 12, it could lead to milder ASD conditions such as Asperger's syndrome or psychotic disorder. And typically in older children, in addition to autism, we also find dystonia and schizophrenia associated with folate receptor autoantibodies. Now, in terms of the effect of antibodies, there are a number of factors that dictate the outcome. And here I'm showing you a diagram of maternal and paternal antibodies. If both parents are positive for the antibodies and the fetus is exposed to the antibody during embryonic and fetal development, the prognosis is poor and the disease is severe. If the father is negative for the antibody and the mother is positive for the antibody, again, this could lead to severe autism because the fetus is constantly exposed to the maternal antibody. And the response to treatment depends on how the early diagnosis and rigorous treatment. If the father is positive and the mother is negative for the antibody, typically this could result in moderate to severe autism with variable prognosis. So therefore, identifying parents and screening them for folate receptor antibodies becomes critical in the prevention and management of autism in the offspring. Now, what if both parents are negative for the antibodies? And this scenario does exist where the child develops antibodies to the folate receptor. And what we do know is the earlier the child is exposed to milk antigen, folate receptor is present in high quantities in animal milk and milk products. So the earlier the child is introduced to bovine milk, and if the child is susceptible to the autoimmune disorder, the child will develop high titers of the both blocking and binding type of antibodies and will develop severe CSF folate deficiency and cerebral folate deficiency syndrome. Okay. Now, if the child is exposed to antibodies, again, milk products early on, that child may not develop high titer of antibodies. It may develop a moderate titer of antibodies and will eventually show up as acquired infantile autism. Again, early diagnosis and treatment has a favorable outcome here. Now, the best scenario is when the child is not exposed to bovine or other sources of animal milk for a longer period, such as a mother breastfeeding perhaps for a year or longer. And in this case, the child develops antibody later on. And if at all, the child could develop a milder form of autism later on in life. So the best scenario here is to try and avoid animal milk and milk products as long as possible to prevent this sort of thing occurring. So in terms of treatment strategy, clearly a milk-free diet and high-dose folate administration would be the ideal thing to do. So the question here is now, what is the folate form of choice? And as pharmaceutical preparations, you have folic acid available, which is a very stable form, readily available and cheap. Then there is methylfolate, which is not so stable, but it is also readily available. And then there is folinic acid. The question is, which form should be used in the treatment of ASD? So to address this question, we looked at folate absorption in the rat. We took adult rats and looked at folate absorption after oral administration. So we used folic acid, 5-methyl, leucovorin, which is DL-folinic acid, only the L form is metabolically active, and fusilib or levoleucovorin, which is only the L form and is fully metabolically active. If you look at the purple bar down the panel, you'll see that no matter which form is used, typical peak activity concentration in the blood is seen around 60 minutes. The green bar shows conversion to methylfolate, and most forms are converted to methylfolate, perhaps more so in the case of levoleucovorin. Also, there is more tetrahydrofolate formed from levoleucovorin. The one thing about using folic acid is that, as you see on the red bar here, there is a significant amount of unconverted folic acid appearing in the circulation. So looking at unconverted compound appearing in circulation, the red bars show folic acid, and there is a very high amount of folic acid in circulation. Mind you, the dose administered here was 4 milligrams per kilogram body weight, which is twice the dose that is highest dose given to children with the autism spectrum disorder. And at this pharmacologic dose, clearly there is a significant amount of unconverted folic acid appearing in circulation. There is some recent literature criticizing the use of folic acid even as a supplement because they feel that the unconverted folic acid has a negative effect metabolically, and it may be detrimental to folate metabolism. So clearly, in this case, where pharmacologic doses are administered, folic acid is not the compound of choice. Methylfolate, on the other hand, can be administered, and it appears unconverted into circulation, as seen here, and it can be readily converted to other forms of folate via the enzyme, the methionine synthase and dihydrofolate reductase. So looking at the next two panels here, D-L-folinic acid and levofolinate, most of the levofolinate is converted to methylfolate. And the advantage of using D-L-folinate or levofolinate is that both forms can be used. Both forms can be utilized in metabolic processes. So even if it is not converted to methylfolate, the form that is not converted can be readily used in metabolic reactions. So to connect folate receptor antibodies to autism spectrum disorder, we looked at the rat model of exposure to antibodies during pregnancy. A rat model of exposure to folate receptor antibodies shows behavioral deficits such as social interaction deficits, learning, memory, and cognition deficits in the pups. And this data is all published, is out there in the public domain. These deficits can be prevented by pre-administration of folinic acid. So what could be the potential mechanism, both of the pathology as well as the rescue? To investigate this, we looked at folate and folate antibody transport to the fetus and to the brain. So shown on the right top panel here is the brown staining material is the presence of folate receptor antibodies, in this case, rat folate receptor antibody administered intraperitoneally, and its distribution in a pregnant rat. So you can see there is a tremendous accumulation in the placenta, in the yolk sac, as well as in the embryo, much less in the embryo. A quantitative evaluation of that indicates that most of the antibody collects in the uterus, and in the placenta, very little goes to the embryo. And you can see higher magnifications of the embryo here, again, accumulation in the embryo as well as in the chamber ventricles and the choroid plexus. Now, what is the result of that? The result of that is if you give now radioactive folic acid to an animal which has received the antibody, what it does is it blocks folate. It blocks folate transport. That blockage is seen most in the embryo. Here's the normal embryo. Here is the embryo receiving folate receptor antibody, 1 or 2 doses, depending on 48 or 72 hours. And that results in a significant decrease in folate transport to the embryo. Let me get this thing out of there. Okay. All right. Next slide. So then we asked the question, is the distribution of the folate itself, in this case, we use biotin conjugated folic acid to try and trace it. And when we gave it to pregnant rats, as you look at the distribution in the placenta and in the embryo itself, this is as early as 30 minutes after IP injection. You can see there is tremendous accumulation of folic acid in the placenta, in the embryo, you see at 30 minutes, there's a lot of accumulation in the heart, in the liver, and also regions of the brain, but much less so. And by 1 hour, there is more accumulation of this in the embryo, and a higher magnification down here shows all that brown staining stuff is now folate distribution within the uterus, the placenta, as well as the embryonic tissue, including the choroid plexus and regions of the brain. So clearly, the antibody distributes to regions similar to where the folate would be distributed, and this could affect folate uptake and deposition within the brain tissue. And when we looked at a young brain, in this case PND22 rat brain, again IP injected biotin PGA distributes to, this is the cerebrum, this is the cerebellum, and the time course ranging from 5 minutes all the way to overnight. And as you can see, as early as 5 minutes, you start seeing the appearance of biotin PGA within the brain, much less so in the cerebellum, but with increasing time, and by 30 minutes, you see a fairly large accumulation of folic acid in the cerebrum as well as in the cerebellum. And this accumulation, even though it seems to decrease a little over overnight period, it seems to remain so much so in the cerebrum. And the distribution is typically seen around the tracts of the white matter in here. And seen in the next slide is a higher magnification of this, showing you distribution of biotin PGA after intraperitoneal injection. As you can see, most of the distribution is in the white matter tracts, in the internal capsule area, the external capsule area, the fimbriae, and in the ventricular chamber in the choroid plexus. Now, if the folate is administered 24 hours after antibody administration, this distribution is decreased. You can see there's no specific area of decrease, but overall there is a decrease in folate distribution within the white matter of the cerebrum. And if one looks at the antibody distribution, you can see there is a high accumulation of antibody in the choroid plexus. Antibody distribution can also be seen in the microvascular, in the blood vessel, in the brain itself. So there is a clear effect of the antibody on folate distribution in the brain. And this could be the reason why the presence of antibody leads to cerebral folate deficiency. Now, one of the questions we have tried to address is the behavioral deficits that we see in pups exposed to antibody during pregnancy, is this transmissible to subsequent generation? If it is, it would indicate that it is epigenetic. There are epigenetic changes. This could be in the methylome, gene expression profiles, and then it could readily be transmitted to subsequent generation. The reason for asking this question is that autism runs in families. But it is not a genetic disorder in the sense that Mendelian genetics is transmitted, but it is a disorder that runs in families. There is a genetic component involved. So we wanted to see if the behavioral deficits that we see in F1 generation, these are the pups exposed to the antibody during pregnancy, would transmit to subsequent generation. And from this preliminary study, whether it is looking at vocalizations on PND4, which is an indicator of bonding between the mother and the newborn pups, or whether it is looking at male vocalizing of bonding in older pups, as well as active and conflict avoidance tests, which are indicators of deficits in higher learning, such as learning, memory, and cognition. Are these present in the first generation, and are they transmitted to subsequent generation? And from this experiment, it appears the answer is yes, that in fact, the deficits that we see in the pups directly exposed to the antibody are transmitted to subsequent generation. So what is the relevance of this experiment? And how could this experiment then lead to subsequently identifying the potential causes of developmental deficits that we see in patients exposed to folate receptor antibodies? Lies in the fact that this animal model allows us to look at the epigenome, the methylome, and also identify the genes involved. It also allows us to basically experimentally manipulate the epigenome and to see if we can change it or restore it so that we can prevent these deficits being transmitted to subsequent generation. These are very important studies that could perhaps provide an answer to the behavioral deficits that we see in exposure to folate receptor antibody and folate deficiency. We are most grateful for the Brain Foundation for funding this ongoing research, and hopefully for the future research, which is going to need a lot of funding to really look at the epigenome, the genome, and to see how we can restore the normalcy to this genome to prevent transmission of these behavioral deficits. So in conclusion, levofolinate has shown great promise in the treatment of ASD. And this is evident from trials that have already been completed by Dr. Fry and Dr. Ray Makers. And with significant improvement in speech, communication, and social interaction. There are at least 3 clinical trials ongoing, and hopefully this will give us more data. I believe that it holds most promise, perhaps in the prevention of ASD by identifying and treating parents prior to conceiving. Therefore, screening for folate receptor antibodies, reducing antibody titer, and providing adequate folate are critical for a positive outcome. Maternal cord blood testing for folate receptor antibodies could provide for follow-up and early intervention in the newborn to prevent development of ASD. So that's our ultimate hope, to see how we can prevent the development of ASD. The last data I heard from the CDC is now 1 in 44 children diagnosed with ASD. This upward trajectory needs to be changed, stopped, or shifted to the downward trajectory. And our hope is that we may be able to do it if we can identify what is the cause at the genetic level that is causing this. I have many people to thank for some of the work that I presented here today. Up top panel is my lab group. Most of the work I presented was done by an MD PhD student, Ankuri here, and Jeffrey Sequeira, my research assistant. Current ongoing work. Oh, how do I get back to this? Jeez, what happened? Okay, current ongoing work is being done by Natasha, my graduate student, and hopefully we will have some answers in the near future. Thank you for your attention.
Thanks so much, Ed. That was really terrific. What a wonderful talk, and it's such promising work. I think Dr. Woody has the first question.
Okay, how do I get back on? I can't see myself, or?
Just stop sharing.
Stop sharing. Let's go back. Okay, great. All right, I'm back. I'm sorry about that last slide. It flipped and I went back to the first slide. Okay, so the questions are in the QA or chat?
They are. Dr. Woody had the first question. Did you want to?
Dr. Woody is in chat or Q&A?
No, he can just say it if he wants.
Oh, okay, sure.
If not, you can start.
So, Dr. Woody, you have a question?
Yes, I do. Are you familiar with the new therapy using anti-FcRn antibodies to eliminate pathologic antibodies in patients, which has actually been used in pregnant women to eliminate anti-red cell antibodies?
Yeah, there are treatments to eliminate antibodies. There are also treatments in using drugs that reduce antibody production. There are also treatments that use IgG, infuse IgG to reduce antibody. These are all general treatments used for a variety of autoimmune disorder. They're not specific for autism related, but they have been used in CFD patients with severe CFD syndrome, where the antibody titers are very high. I'm not aware of anything that has been used to specifically remove folate receptor antibodies.
Yeah, the rest of the questions are in the chat. Do you have the chat, or you want me to read them to you?
Would you read them to me, Richard?
Okay.
That would be most helpful.
So the first question is about the study that showed high doses or high levels of folic acid in the blood maternally has been linked to autism. And the question is, how would a too high dose of folic acid in the pregnancy lead to increased autism? Would it be through in vitro hurting the child, or through an autoimmune reaction of the mother?
Okay. Most of the studies reported, they did not involve pharmacologic folic acid administration. They were in the general population showing small amounts of unmetabolized folic acid. My explanation for that is, if you're on a daily B complex or multivitamin that includes folic acid, you're likely to see some unmetabolized folic acid in circulation. There could be some unmetabolized folic acid intracellularly also, because the enzyme dihydrofolate reductase is not able to very efficiently metabolize all of the folic acid. So if the concentration drops below the km, you're going to have some folic acid in the circulation. Whether it causes autism, I'm a bit skeptical about that. I'm not sure. I'm not buying that theory, even though they have shown that there is some link statistically between these two.
Okay. Next question is, given the transgenerational influence, at what gestational stage is the FRAT most beneficial for prenatal testing?
Ideally, the FRAT should be used in parents before conceiving. If both parents are positive or one parent is positive, they should be treated both to reduce the antibody titer and to provide adequate folate. That would be the ideal scenario, where the parents are screened prior to conceiving. And if that is not done, then I think introducing larger doses of folinic acid during pregnancy and also during early childhood may help prevent the disorder, those who are susceptible to the disorder.
Okay. Now, an interesting question: what is the range of epitopes? What is known about the immunogenicity of the bovine folate receptor, that is immunogenic polymorphisms? Any info on different strains of cows? Have you looked directly at the methylome in rats in high titer or in high titer humans?
We have not looked at the methylome in the rats or humans, but that is clearly our objective to going forward, to look at the epigenome in some detail and to see what changes have taken place, and can we restore these changes by providing folinic acid to the rats before pregnancy and during pregnancy, and see if the outcome changes. Now, what was the first part of the question?
Whether there's any epitopes specifically. Is there, do you know anything?
Yeah. So what we do know is that bovine milk is highly immunogenic. So is camel milk folate receptor. The least immunogenic is actually goat milk, even though goat milk contains about twice as much folate receptor as bovine and camel milk. Okay. Epitope-wise, the antibodies that we have identified are blocking type. So they bind to the active site of the folate receptor, whether it is irrespective of which source the antigen comes from. And then there are binding antibodies which identify epitopes that are distinct from the folate binding sites. All of these antigens have greater than 95% homology in terms of amino acid sequence.
Okay. And one more question we have time for. Is there any relation between maternal age and FRAT?
Maternal age and FRAT. Good question. I don't know the answer to that because we really haven't looked at age-wise. I can tell you this, that when we've looked at women with neural tube defect pregnancy, these women mostly were in their late 30s and 40s. They've had an NTD child maybe 8 years earlier or 10 years earlier, but they still were positive for the antibody. So once you have the autoimmune disorder, you're likely to be positive for a long time. Now, one thing that reduces the titer of the antibody and eventually gets you to negative area is if you go on a totally milk-free diet, as was the case with one mother who, we have published this, had repeated miscarriages and had difficulty in conceiving. She had a high titer of the antibody, and she was put in a milk-free diet for over a year. And at the same time, she was monitored for this antibody titer. And eventually, she became negative for the titer. She conceived normally, carried to term, and had a normal child.
Super. Well, you can answer the rest of them in the chat, Ed.
Okay.
You can get to it, and we can move on to our next speaker.
Thank you.
Thank you, Ed. All right, so it's my pleasure now to introduce Mentioned Child Find program. He's been the PI of several NIH and NBHB funded studies of interventions to improve early development. He's the head of the LEND program at SUNY, and he has many interests. And with that, I will let him give you great information about his work on folate antibody and trajectories of autism in early childhood identification and treatment. Do you have your PowerPoint? Yeah, great.
Sorry, can you all hear me? And
yeah, just from the beginning. Excellent.
Okay. So what I'd like to do is to go over very, very quickly, because I think Ed has covered it, an overview of folate receptor autoantibodies, the linked ASD, use of leucovorin. I'd like to show two clips of a before and after of a child we treated, a 4-year-old with leucovorin, looking at, and then look at an objective measure, the BOSC. I'll talk about that in a minute, to look at how you objectively measure social communication change. I'd like to come back to the questions that Ed raised about postnatal and prenatal impacts, and then finally move to how this is the rationale for our current Brain Foundation funded study. So let me start. Just to say, I have no financial interest in any of these. We will talk about non-FDA approved use of leucovorin. So can I just say, as a pediatrician, I've worked, our clinic largely sees children on the spectrum at different ages, and we worked for a long time. We see it as a complex, multifactorial process. So when a colleague said, introduced me to some of this work, and Ed's work and Richard's, that, oh, autism, it's due to a lack of folate in the CNS, I must admit that my reaction was this. It sounded like something out of a tabloid. It sounded like, oh my goodness, autism cure found and it's a vitamin. And I was skeptical, to say the least. I've come around. If I could just go over, there's some very convincing evidence on this, that folate receptors disrupt the folate receptor alpha, that it results in CNS folate deficiency, the strong link to autism spectrum, that you certainly see a very, very high rate of, in children diagnosed with ASD, this would be comparing to folate receptor positive rates, maybe of 10 to 15% in the general population, depending on the study. But most strikingly, and this is especially work that Dr. Fry has done, that treating with leucovorin, bypassing the folate receptor blocking step and correcting central nervous system folate, really shows a clear clinical improvement, certainly in language, but also in social communication in preschool and school-age children. So let me start by seeing if I can get this to work. I'm going to show a 4-year-old, sorry, if I could just go back. So this is a 4-year-old with autism spectrum. He had language delay, very poor social interaction, and we arranged the testing through Ed's lab and he was positive for blocking FRA. So let me show, if I just open this.
We have more race cars. Let's open this. Let's see what we can do with this.
Can I just ask, are you all able to hear that? Was that okay? Was that working?
Yes.
Yes, yep.
Ooh, that's so cool. One for you. Look, and one for me. Should we race the cars? Let's see what this does. Put this down. Oh, good job. Can you turn this? You turn it. Can you turn it? Yes, good. What is that? What animal is that? That's a chicken? Good job. What else do we have? Oh, another one. Ooh, let's do it together, Nikos. Let's see, can we do it together? You do one and I'll do this one. Look, which one do you want to do first? You pick one here and I'll do the same one here. Okay, you want to pick this one? Oh, you want to do race cars? Ready? 3. Look at this. Okay. Ready? Should we make it go? Pull it back. There it goes. There goes Nico. Look. There it goes. Oh, there it goes. You want to make yours go? Here, put your guy in here. Pull it back. Here, look. Pull it back. And go. Doesn't quite go. Here we go. Ready? 1, 2, 3. Go! Whoa. What else do we have in here? What is that?
He loves music, so those triangles will probably be right up his alley.
Oh, look! Dump truck. Did you want to put some blocks in it? Yeah, look. We have the blocks in the dump truck. Oh, that's a dump truck. Let's put some blocks in it.
Okay, I think you get a flavor. Okay, so we treated him for 6 months with 2 milligrams per kilo of leucovorin, and this is when he came back.
Okay, look at Nikos. Look. Which one should we open? Can we do this together? Which one should I open? You want to open number 1? I have a chicken also. Is yours not closing?
Honey.
There we go. You turn that.
1, 2, 3.
Wonderful. Now you open yours. Same way.
What's this?
Turn that. What is that? What animal is that? A chicken. Yes. Who's under 2?
It's crazy.
Who's that? What animal is that?
A chicken.
Under 2. Under number 2. What is that? What animal is that? Open that. That's a sheep. Open number 3. You got to turn it this way. Like that. Turn it. Yep. Oh, I think you do. There you go. Who's that?
Open.
What animal is that, Nico? This one. That's a horsey. That's a pig. Should we see what noises these animals make? Look. I have a farmer here. See? And where does the horse go? Can you tell me where these animals go? Tell me where these animals go. Look. There's a picture of the animal right here, and then you have to match the animal. Can you match the animal with the picture? I'll help you. What animal is this? Where does pig go?
Right there.
Yeah, that's right. And then it makes a noise. You want to push him down? Push him down. That's a cow. That's the farmer. That's a horse. Where's our sheep? Did we lose our sheep? Oh, here she is. There we go.
She's right there.
Yes. And if you press them down, you'll hear the noises they make.
That's pretty impressive, Harris. I think maybe we move on in the sake of time.
Yeah, do you know the song? Oh, MacDonald had a farm. I'm not working. It's not working. So what should we do now?
Thank you. Okay. So, so we used a measure called the BOSC, and this basically is a brief observation of social communication change. I don't want to go through the algorithm. The point is that for this and for about 15 different features, it really operationally has you score. It's derived from the ADOS by the same authors, with a low score being a more typical child, a higher score being more autistic. So on eye contact, this child went from baseline of 4 before and now scored a 1. On social overtures, he went from a baseline of 5 down to 3. If you look at the overall scoring, if you add these all up, he first of all, if I could just ask clinically, is this child still on the autism spectrum? I think the answer is yes. But with an overall score going from 39 down to 19, I think it's consistent with both the parents telling us and our own impression that this is a much more cooperative, engaged, and responsive child. Okay, so this led to proposals that we could ameliorate ASD by early identifying and treating it. But I think the treatment strategies, as Ed started talking about, will depend on understanding what's going on with FRA. This, as Ed covered, are we talking about sort of postnatal exposure to milk, for example, triggering some degree of titer and leading to this, and then we would look in the pediatric period to identify and treat to reduce and autism. On the other hand, if we're talking about parents who are positive and this is being transmitted, and especially when the mother is positive, then you want to look at screening mother and father and looking at obstetric care. Why does it matter? This is a work from Dr. Raymakers. I think he presented last year. Basically, this is the CARS score, where anything over 30 would be meeting threshold and the higher, more autistic, comparing blue baseline he had a range of kids of different ages, but then treated them for 2 years, and this would be the score after 2 years of leucovorin treatment, and where K is the positivity of the child and the mother, P the father. So if you could just look at the difference when the child is positive, but both parents are negative, the baseline, first of all, across is somewhat lower. The improvement with leucovorin much more striking. At the other end, if the child is positive, both parents are positive, they're starting out at a much higher baseline, and in fact, the improvement is slight and not significant. So this matters. As Ed said, FRA positivity runs in families. That something like 60 to 70 in one of his studies of parents as well as non-affected siblings were also FRA positive, raising the question, why is it that if you're FRA positive, why do you sometimes get a picture of ASD in some individuals and not in others? Okay, so I think the answer clearly has to do with timing of when the FRA is turning positive, as well as the interplay of many other factors, metabolic factors, genetic factors, and exposures. As far as I know, though, there have not been longitudinal studies. The studies we've heard so far are cross-sectional, looking at where a parent or a child is long after a child is diagnosed, but there haven't been studies starting at birth and looking forward of FRA status in the parents and the child in terms of their ASD development. And so the question we are asking in our Brain Foundation study is, if we were to examine the trajectory of FRA status and looking at later diagnosis of infants and starting with these are infants at risk for spectrum based on already having parents coming in with an older sibling who is positive, could we tease apart the contributions of FRAs that are present in the newborn, as in prenatally, from those that develop later in infancy, and how does that correlate with the emergence and severity of ASD features in subsequent years of life? Okay, so the goals of our Brain Foundation and O'Sullivan Foundation grant study, first of all, to look at the trajectory of FRA status in cord blood, and then at 6 and 12 months, and compare that to the clinical course. We will try very hard to go through the second year of life. The grant period is short, but to look at when and how much do symptoms and a diagnosis of autism emerge. Secondarily, to look at brain function, to use FNIRS, functional near-infrared spectroscopy, which is a non-invasive neuroimaging, as well as what's more classically been used is eye tracking, as early biomarkers to look for, even in infancy, and there's studies on certainly in eye tracking and some in FNIRS suggesting even before autism emerges, you can see differences in brain function, response to social stimuli versus not. And if we can then see how they correlate with FRA status and eventually a later ASD diagnosis, the overall goal would be to pave the way for studies using leucovorin treatment and in infancy and eventually prenatally. So how we're doing this, we'd like to enroll 30 to 35 pregnant mothers who have a previous child with autism spectrum, who are going to deliver at either Downstate or some of our affiliated hospitals, to determine the status, FRA status in cord blood, we're figuring we're at each step going to get some loss to follow-up. Following this cohort through the first year of life to look at ultimate diagnosis with ASD, measuring FRA status at 6, 12, and 24 months, in addition to looking at a whole series of environmental factors, toxic exposures, as Dr. Fry talked about, and especially milk and bovine milk exposure. And then to look at their cognitive as well as social development at 6, 9, and 12. There are not a lot of measures in the first year of life. One is the communication symbolic behavior scales. The Mullen is used for cognitive and language. We would also use the FNIRS and eye tracking. And then the idea would be to continue them into the second year of life when you can start to use more classic gold standard measures such as the ADOS, the ADIR, the Autism Diagnostic Inventory, and continue with the. So in conclusion, if we could better understand the child's FRA trajectory, the ways it's influenced by the parents' FRA status, environmental factors, how these correlate with manifestations of ASD and brain function. Could we then better design treatment and preventive interventions, including what kind of approach? Are we starting in infancy, or should we really need to start in prenatally, as Dr. Quadros? So getting back to is a vitamin going to cure autism? I think probably not. But is folate treatment, can it make a major difference in ameliorating and improving functioning in very large subsets of individuals with ASD? I think so. So thank you very much.
Thanks so much for that great presentation, Dr. Huberman. We're supposed to have a break. We're supposed to have a 15-minute break, but I would propose that if you could take any questions in the Q&A, through typing in, and maybe we take a 10-minute break and come back at 11:05.
Yes, that's
All right, that sounds great. Thank you, everybody.